Friends, today we are again started with our fifth tutorial video, that is all about auto pipettes. So we are now going to discuss the next very important instrument, that is after incubator. There's a very important instrument that is for transferring the media. This instrument is used for transferring the media, the culture media, the fungal media, for slide preparation, and any minute transferable thing, to move any transferable thing from the culture media to the flask, or anything, any form transfer for all types of transfer, liquid transfer. We need this auto pipette. Now, what is or pipette? Pipette is just, if you have heard in the chemistry laboratory, you have seen a glass pipette, a glass dropper. Those are not automatic. They used to use this is the pipette. This is a standard pipette. As you can see in front of me, it is a standard pipette. This is a standard pipette. This is a standard glass pipette. It is graduated. You can easily measure. Here, the end is all about this end. All is about the end. Difference is this end. Now, what you used to do in this pipette, what we can do, either you have to put a rubber bulb like that, rubber bulb here, or you have to suck it with the help of mouth. And in microbiology lab, sucking the culture media and sucking the parasites and the bacteriological media, or the fungal base or bacteriological base or any base, it is very dangerous to suck by mouth or sucking by a rubber bulb. Both are dangerous. So we need a special type of sucking device, which a piston device. It is a piston device which will press and leave. So it will suck. It will draw out all the air from the pipette and it will suck. When you release, it will suck, release the plunger, it will suck. So that is the system of auto pipette. And another thing is that you can measure like this. That is also a measuring device and more accurate than this. Here you can miss. If you suck, you can suck more or you can suck less, according to your mouth, the sucking power of your mouth, or the sucking power of the bulb you are sucking. So you can suck more or you can suck less. So that is not done in the auto pipette. Auto pipette is very definite. And also that you can suck from 1 ml. No, you cannot suck less than 1 ml. You can suck less than 1 ml from it. There is another auto pipette. You can suck less than 1 ml, but not less than 1 micro ml. You cannot suck 1 micro ml, or you cannot suck 2 micro ml, or you cannot suck 20 micro ml, or you cannot suck 65 micro ml. That you cannot suck. It is very difficult. It will be very difficult. And that type of precisions you cannot make in glass. So therefore, we are using this type of auto pipettes. We have 4 auto pipettes here. Just you will see in the next video, next part of this video, you will see there are 4 auto pipettes we will be discussing all about. And these auto pipettes are specially designed and they are used in microbiology. So without these auto pipettes, you cannot proceed in microbiology. So we are now going to discuss with these auto pipettes. So how to use properly, how to properly use these auto pipettes, and how to hold the auto pipette, how to use the auto pipette, how to put the tips in the auto pipette, what kind of tips you can use, how the tips are disposed, how the tips are used, how the tips are kept. So those are will be discussed in the later part of the video. That is going to be an interesting video for you, because auto pipettes are purchased by many labs, but I can say that very few labs or very few person can use properly the auto pipette. How much auto pipette is there? How many types of auto pipettes are there? It is nobody knows fully. So that detail I will be discussing in this later part of this video. So be with me to see the tutorial video of auto pipettes, all about auto pipettes. So just stay with me. So friends, you can see, and this is the auto pipette. I have 4 auto pipettes in this camera. You can see there are two auto pipettes are here and 2 auto pipettes are here. So first of all we will be talking about this this auto pipette. So this is a 1 ml variable auto pipette. This is from 10 micro ml to 100 ml auto pipette. 1000 micro ml auto pipette. 1000 micro ml means 1 ml. 1 microliter means 1000 microliter means 1 milliliter. So that you have to remember. Microliter, we are talking here about microliters. We are not talking about milliliters. We are talking about microliters. So that you have to be careful. This reading this unit you should be careful. This is from 10 microliter to 1000 microliter. That you should be careful about. So what I was talking about, that this is the plunger which will press and set all the air outside and release the plunger. It will keep up the liquid. Now this plunger is to strip off the tip. As you can see, the plunger is this steel bar is moving down and it is just moving out the tips. So this is the plunger. So this is the tip releasing plunger, and here you can see a dial. This is a dial and this is the knob, so by which you can move this dial, and the dial you will able to just set the volume of the auto pipette. So this is now, it is 500 micro ml, 050. So this is 500 micro ml. Sorry, 50 micro ml. Sorry. This is 50 micro ml. So now, if we move on, so 70 micro ml, and we moving on, and again now you're increasing. So now this is 100 micro ml, 1000 micro ml. Now this is 1000 micro ml. 100 means 1000 micro ml. So here we're now getting as 500 micro ml. So like this is 900 micro ml. This is 900 micro ml. Like that you can set. So this is now getting 800 micro ml. So we're going towards 800 micro ml. Now this is 700 micro ml. This is 700 micro ml. Like that you can set. So now we're going to see how the auto pipette, the tip is filled up. So here is the tip box. So let's remove this auto pipette, so we will see it later on. So now you're discussing with this one. So this is the tip box. You can see in the front of the camera, this is the tip box. Just open the tip box like this, just like this, and take the auto pipette like this. You have to hold the auto pipette like this. Holding the auto pipette with this, just the forefinger on the top and the 3 fingers below, and this is the thumb. So thumb will push the plunger. All other fingers will be like this, and this is how to hold the auto pipette. This is the only way to hold the auto pipette. Don't hold like this, or don't hold like this. You always hold like this. Your thumb will be, your forefinger will be here, your middle finger will be here, your ring finger will be here, and your little finger will be here, and your thumb will go on the plunger and also on this release button. This, you have the thumb will move. So first you have to place the auto pipette on the tip firmly. Don't make it loose. Don't make it loose. Don't like this. No, not like this. It will come off. So it will fall off. So you have to press the auto pipette firmly and take it out. Now to release, just release this button. And when you will go to put the liquid, first push the plunger in the air. You push the plunger in the air. Don't put the plunger in the liquid. You push the plunger in the air, and then dip the tip inside the plunger, inside the liquid, and slowly release. The tip will take the liquid out. So which I am going to show you now. So let us open this distilled water bottle. So here is the distilled water bottle. So we are going to release this. Take out, and now what we are going to do, just you can see, just we can remove this box, and in the camera you can see. So this is our box. So now we are going to take the pipette tip, just engage the pipette tip, and just press a little bit and take out. So it will coming out. So now what we are going to do, we have to push the plunger and dip inside the bottle and slowly release the plunger, and the liquid is up. Liquid is up. You can see the liquid is up. Now just how to release it? Go inside that and slowly release it. Push the plunger and the liquid will be transferred. Now take it out. So this, don't do this. Don't do this because you will let all the air bubbles inside. So don't do this. You first push the plunger and take out. So this is how to use the auto pipette. Use how to. Don't fully dip the plunger. Don't fully dip the tip. You just dip the tip little bit. Just mouth of the tip. Mouth of the tip. Just push it. Put the mouth of the tip inside and slowly release the plunger. And whole liquid is inside. So this is how to use the auto pipette. So now we are going to see the other auto pipettes. So this auto pipette is over. So we are now going to see other auto pipettes. So this is a small auto pipette. This is the same, just everything is same. So this is our plunger. This is the tip releasing plunger. This is the tip releasing plunger. This is our volume plunger. So how we can take up the liquid. So this is our volume plunger, and this is how to hold auto pipette. Here is a dial. This is from, you can see in the camera. You can see this is from 20 to 200 microliter. It is clearly written over there. So 20 to 200 microliter. So this is from 20 to 200 microliter. You can set this dial. This plunger you have to set. So this you have to just push and just release, and it will be turning. And just push and release, and this is turning. So now it is 200 microliter. Now this is 200 microliter. And then you can set again and just release, and you can just remove this plunger, this knob below the plunger, and you can desirably set the micropipette. So this is 120 microliter. This is moving, and this is 550 microliter, 60 microliter, 70 microliter, like that. You can just set. So this is our auto pipette. So another auto pipette is there. So this is a very small auto pipette, and this is the biggest auto pipette. This is the same way. It is 5 ml auto pipette. This is 5 ml auto pipette. So 1 ml to 5 ml auto pipette. Now this is the big one. We are talking in millimeters now. 1 milliliter to 5 milliliter. This is auto pipette, and the tip is very big. So here the tip is very big. So when you put the tip inside, so this is the tip, and here is the tip. So this is a very big one. This is used to transfer big solutions, big amount of solutions. The same way, it is the same way. It will works in the same way, same auto pipette. Just the volume is different. Just push the plunger as before, put inside, and take the liquid. So the liquid is up. But here one thing is there, that you cannot, there is no automatic release. This auto release button is not there. So you have to release by your hand. So this auto pipette does not contain any plunger release, because this is a very big tip. So you can release by plunger. So you have to remove by your hand. So this is the only difference between the two auto pipettes. So this one and the other ones. This one and the other ones. This one and this one has only one difference, that this plunger button is not present over here. This has the push delivery button or volume button, but volume plunger, but not the tip release plunger. Others are same. This knob is same. This knob is same. Just calibrate the volume changing knob is same. Everything is same. But this tip release plunger is not there. So that is the difference between these two big, 1 ml to 5 ml auto pipette, and this 1 microliter to 1000 microliter or 10 microliter to 1000 microliter auto pipette. So these are the two differences. So another auto pipette is there. This is a fixed volume auto pipette. This is also a 1000 ml micro pipette, but this is fixed volume. There is no volume plunger, volume changing knob. It is only a plunger changing knob and a tip releasing knob and the plunger, volume plunger, just a pipetting plunger, or you can say the volume taking up plunger, release and take up plunger. And the same thing is that pipette end, tip is end is that the same 1 ml pipette tip is will be using. So this is all about. So one thing you should be careful about this pipette, auto pipettes, that 3 things you have to remember. First, engaging the tips, then really taking up the volume. First you have to release all the air from the pipette tip, and then you dip in the solution, and then slowly release the plunger. Don't put air bubbles in the media, because what will happen that if you put the air bubbles, if you insert air bubbles in the fungal media, the fungal media will be damaged. The fungal cells will be ruptured. The bacterial cells can rupture because they will get an air embolism. So they will rupture. So never ever release the air inside the media. Always release the air outside the media. And then put the plunger, then put that auto tip of the auto pipette, and just slowly release the button. And the pipette will take up the liquid. Whatever the fungal base or whatever the matter, whatever the volume you want, that you can release. Another pipette is there, which I have shown before. This is Pipette. So this is also a fully powered auto pipette. This is a battery powered auto pipette. This is an also auto pipette, but this is, you cannot fix the volume. It is battery powered. It is same like the rubber pump which we are using, same like a rubber pump, but it is a controllable rubber pump. You can with these 2 shafts you can control, these 2 switches by which you can control, because this motor speed is a motor inside, pump inside. So this pump actually pushes the liquid. So when you insert a pipette like this, a pipette is there. So this is a 5 ml pipette. You can, if you insert this 5 ml pipette over here, this is also an auto pipette like that, and it does not release air. It does not release air. So it is not required. You just put inside, and you have to switch on the power. Before that, the power is off. So now, and it is taking up the water, liquid, and you can desire volume. You can just stop when the volume comes, when the pipette tip comes, when the meniscus of the liquid comes. You can take the volume. You can make more. You can increase up to the 0 mark. You can take up to the 0 mark and stop. It is very easy. So this is a and release button, and just release and it will go, and the liquid will go. So this is a very useful pipette, but this is a little bit big, so it is very difficult to use. Generally this is used also, but auto pipettes are more used than these Pipettes. Pipette is also used. Just you have to keep it in the just keep it in the sterile enclosure. And this is the auto pipette. These are available. The sterile auto pipette, sterile pipettes are also available, and these auto pipette tips are also available. They are available in the boxes, about 1000 tips or 500 tips or whatever. It is available. So one tip is 5 ml auto pipette. Another tip is 1 ml auto pipette. For 1 ml to up to 1 ml auto pipette, 1000 micro ml auto pipette, and another one is up to 200 micro ml auto pipette. So these 3 types of micro tips are generally used. As you can see in the camera is showing. As you can see, these 3 auto pipettes are generally used. You can see when I just move the camera towards it, and you can see this is the 200 ml auto pipette, this is 1000 ml micro ml auto pipette, and this is 1000 ml micro ml auto pipette, and this is 5000 micro ml auto pipette, or 5 ml auto pipette. So these 3 types of tips are generally used in the microbiology lab. So, and the fourth one is the Pipette. This is a 5 ml Pipette. This is a 5 ml Pipette. So these 4 types of auto pipettes are generally used. So this is all about, this is for the 5 ml. Here you can place, and this is for variable and 1 ml, and this is also another 1 ml you can use, and this is the 5 ml, this is the Pipette, and this one is for 200 ml micropipette. So this is the full set of pipettes. You can see in the camera. If I can move the camera a little bit back, and you can see in this camera the whole set is, I think it is coming, the whole set is coming. Yes, the whole set is coming. Yes, the whole set is coming, so that you can see inside the camera. So all about the auto pipettes, and signing off now. We will be soon with the next video about autoclave. Now that video will be very important. We will be regarding about the distilled water, and then we will be talking about sterilization and disinfection. First we will be talking about the distilled water, which will be used for sterilization, and we will be using for disinfection, and then we will go for sterilization. So these three parts are very important. The distilled water, the disinfection, and sterilization. That will be our next video. So there we will be using two instruments which I will demonstrate. So one will be autoclave and another one is UV chamber. So that we will be discussing in the next video of the next tutorial. So thank you from now.